walks and stuff around town.
It seems like a fun.
Yeah. It'd be cool to go see him.
Yeah. It doesn't seem like he's done it since like last year though. At least with
Okay, so what we're going to do for leading into up this next exam, lecture 3 exam, I mentioned this is going to be kind of a shorty. It'll probably only be about 4 or 5 pages long. But that's okay. It'll make it a little easier for you. We're going to finish out talking about muscle physiology, skeleton situations that happen in the animal skeleton considerations, and then nerve cell physiology and nerve evolution. So we'll have kind of all these topics piled in here. And so that's why I wanted to jump gears and get this done now. So obviously muscles, or the ability to move, is a major thing for animals. Even from the very beginning, when we looked at Nigeria, the jellyfish and the anemones, they have muscles. Even though they don't have mesoderm, they have striated muscles that use to move their jellyfish body or capture prey with their tentacles, etc. But then very quickly we see mesoderm arise in flatworms, and when we talk about muscles, it's mostly mesodermally derived tissue. So anyway, I do want you to kind of get an idea about the different types of actions muscles can have with skeletons. We'll talk about skeletons on Tuesday. But muscles basically, okay, the main important thing here is that a muscle, if you look at kind of how a muscle is organized, muscles are an organ basically made up of protein filaments. Okay, so we have our muscle body here. This is kind of like the body of our muscle. We're talking about skeletal muscle here to start with. We'll cover smooth muscle maybe on Tuesday. But anyway, oops, let me go ahead and use too many color choices here. So, we have our tendon, or there. Anyway, so the main thing to note is we have muscles are made up of protein fibers that are kind of bundled together. And the main thing I kind of want you to get here is that muscles can only really do one action. Is muscles can contract or shorten. So a muscle really only has one possible thing it can do. The muscle fibers can shorten or contract, and that's where their power or strength is. They can't extend themselves, and they can't broaden themselves. It's really only one action they can do, which is shorten. So this contraction action is a shortening. That being said, most skeletal organisms are made up of opposing muscles. So, for example, on your leg, you have your hamstring, or your muscle behind your leg, kind of attached to your pelvis and the back of your knee, and that's your flexor. You have a flexor, or oftentimes I already called it an adductor. So the idea is that flexion, what that means basically is flexors, or sometimes called adductors, these bring your limbs closer to the center line of your body. So flexors or adductors are usually drawing limbs closer to your center core, or your center of your body. So your hamstrings are flexors or adductors because they're pulling like your foot closer to your body, or your center of your. Same thing with your biceps. Biceps are pulling your arm closer to your midline. So that's a flexor, or the muscles themselves are called adductor muscles, but the action is called flexion. The opposite, which I think would be important to know, this would be like a good compare and contrast question, flexion versus extension. And the reason why I think it's important is because extensors are muscles that push your limbs away. And this term is called abductors. And I know this is a really rough, and some people had a rough time with this in the test, but it's easy to remember because adductors means you're adding things, you're pulling it, putting them together, right? And abductors, well, I don't know what to say about that, but it's basically the opposite. So abduction is where you're pushing or extending your limbs away from the body. And this is important because if you think about, and we'll talk about this with skeletons on Tuesday. So in extension, we have the limb being pushed away. But the main thing I'm trying to get at here is that we didn't really see organisms that had extensors or abduction until we saw skeletons. The annelids, the earthworms, had a hydrostatic skeleton where they could squeeze their circular muscles and the coelom and push their head out. The flatworms and the cnidarian, the jellyfish, could only really pull their body closer to the middle. They couldn't really push their limbs out. For example, like jellyfish can't shoot out a tentacle and grab a prey, right? They have to basically let the prey drift into their tentacles. So the other thing to note here is oftentimes mussels will have what's called a opposing muscle. So because I said that these muscles can only shorten, when you contract your flexor, it will actually extend or stretch the opposite muscle. And the same holds true for the extension. When you contract your extensors, it will stretch your opposite muscle. So what you find in organisms with skeletons is because muscles can't extend naturally, they basically can relax. They get stretched or pulled by an opposing muscle. So we'll discuss that more with skeletons, but it's kind of an interesting feature. I think the main thing I want you to understand here is that the only action, this is one of the study guide questions, the only action a muscle can do is shorten, contract. Okay, so let's go ahead and, like, as if we were looking at a cross-section of a muscle. Muscles are basically made of bundles of fibers. So we have bundles of fibers that kind of go together. So if I were to draw this muscle here and cut it in half, let's say I were to take a knife or something and kind of chop this muscle in half, what you're seeing basically is bundles of muscle fibers piled in here. Okay, so there's our muscle fibers like that. And then if I look even closer, what you're seeing here basically is the muscle fibers are organized in a very, very specific way. Let me go ahead and do this here. Let's see here. So these fibers are made up of there's two basic kind of working proteins. There's about a half dozen proteins that we're going to talk about. But the two major proteins that we're talking about here are called actin proteins and myosin proteins. Let's go ahead and draw kind of a model of how this goes. Okay. So what we're talking about here is these little fibers that are sticking out here. If I were to pull the fibers out, we saw this on our skeletal muscle, like on the exam. We saw the fibers kind of running like that, and then we saw that kind of artifact, or the cross-hatching on the fiber. This is called striated muscle. And the striations, the little cross striations, so the fiber part is our proteins that are going parallel here, but the cross stria are caused by these end caps. The stria are actually caused by the organization of the muscles. And these are often called Z-lines. And Z-lines. And I think the reason why they're called Z-lines is when you're focusing in on that, they kind of have like a little, now I'm going to exaggerate this, they kind of have like a zigzag feature. So those crosshatch lines kind of, they're not like a straight line crossing the parallel muscle fibers. They kind of go like a little bit of a wavy feature. So the name Z-line comes from the fact that these lines sometimes wave or zigzag as they go across the muscle fibers. It kind of gives you an idea where the name comes from. Okay, so we're looking down here at like the submicroscopic level. You knew that this was like at 450x, you can see like this level of detail. What we're talking about here is really down to even smaller than an electron microscope can see. So this is kind of a model for how muscles work. You actually can't see this. It's at the subcellular level. So we're talking about like, 1 micron or so in size here. Okay, so these Z lines are basically making up what is called the sarcomere. The sarcomere literally means, sarco means flesh, and mere means unit. So this is called the flesh unit, but what it really refers to is the contractile unit of the muscles. They probably call it the flesh unit because, when you eat meat, when you're eating meat in your diet, you're basically eating muscle, right? Chicken breast or steak or whatever. You're basically eating muscle tissue. The name sarco kind of refers to flesh or meat. So it's kind of like the contractile unit of the muscle for the sarcomere. But that's, on the exam, I'll have probably a diagram like this you're going to have to label. And I'll ask you to identify what the sarcomere is. And the sarcomere is basically Z-line to Z-line. It's kind of this is the contractile unit of the muscle. Okay, so there's two basic kinds of proteins here. The red stuff I'm showing here is called actin. Actin protein is a fundamental contracting protein, and in fact, even protists like amoeba. Amoeba, like we talked about how amoeba use their pseudopodia to surround their food or to crawl. Amoeba use actin for cell motion. So actin is kind of a fundamental, basic cell motion material. In fact, I believe, like in cell division, the little fibers that branch out from centrioles to pull chromosomes around are related to actin. So actin is kind of like a fundamental movement protein in organisms, even non-animals, protozoans, and maybe even fungi and plants. So actin is the protein that is on this side, the kind of the parallel fibers here. Actin is sometimes called the thin filament. It's kind of a thinner, there's more of it, and it's a thinner protein, which is why I'm kind of drawing like a little stick here. The other fiber that's kind of on the inside is called myosin. And myosin is called a thick filament. It has a kind of a bundle of proteins that make it kind of thicker in cross section. Okay, so this is myosin. Okay, myosin sometimes is called a thick filament because I'm drawing it kind of like combined together bundles. It's a thicker material, and it kind of lays in between these little fibers of actin. At the very end of the myosin, it terminates into these heads, like this. There's more than what I'm drawing. There's like lots of them. I'm just drawing like a half dozen or so on each side. Myosin heads. Okay. Okay. Okay, so these are called myosin heads. And they're basically at the end, each end of this myosin thick filament fiber, and that's how they terminate. So the myosin is, it's individually the myosin is kind of a thin protein, but it bundles together into this kind of massive bundle. And these heads kind of stick off. And you can kind of think of the myosin heads like little hands, and they're going to basically grab onto the actin and pull it. Okay, so that's kind of how it works. All right, so that's kind of the fundamental parts of the muscle. So on the exam, I'll probably have like a diagram that shows you this and this, unlabeled, and you're going to kind of have to tell me, or you will have to tell me which part's the actin, which part's the myosin, where are the myosin heads, what's the sarcomere, what's the C line. These are kind of all the fundamental parts of how a muscle does this thing. Okay, so here's how it sort of looks. This is what I've drawn. Just showing you one contractile unit. I've shown you three here. But anyway, what happens basically is the actin is kind of like the stick, and the myosin is like my hand. The myosin heads reach over, grab onto the actin, and pull. It's just a little short way. And then they do it again. So basically the muscle contraction action is this kind of the myosin heads crawling down or pulling on the actin filaments. I kind of think of it like tug of war. If you ever think tug of war, you have a rope and you're holding onto the rope and your friend's holding onto the rope, and then you kind of try to use your hands to crawl up the rope. You kind of think of that. That's how the myosin and the actin sort of interact. So the myosin heads, when they branch over and grab onto the actin, it's called cross bridging. The myosin head grabs onto the actin and chemical bonds temporarily, and the myosin head pulls. So we have a very, very specific sequence of events that go on for muscle contraction. And this is how it looks in the book. The book has a similar diagram in the book. But muscle contraction can be thought of as like a cycle. Just kind of bear with me here. It can be thought of as a cycle. The general tendency, the human nature, is to think, where does this start? And I would wager that if I were to ask you where the starting point was, all of us would go here, right? 12 o'clock. I mean, it's just like we naturally think, okay, everything starts at maybe this point, I don't know. That's how I think. Is if you look at a diagram like this that has a cycle, you naturally think the starting point is like the upper one. This would be like number 1, 2, 3, 4, whatever. But I'm going to actually say, suggest that you think, for one, there really is no starting point. It's a cycle, because in order to be here, you have to have had this first, this is next, right? It's a cycle. But really, I'm going to say that our starting point is here. And, I'm not going to put a number on it, but what I'm going to say is the ability for a muscle to contract starts with ATP. And ATP is that rechargeable battery of the cell. It's why you breathe oxygen. You have sugars in your cells, and if you have oxygen, you can make 1/3 of that sugar energy into ATP in your cell. I've mentioned for the last test that animals need lots and lots of ATP because, for example, muscle contraction has a huge ATP demand. Nerve cells have a huge ATP demand. So animals need a lot of ATP in order for their muscles and neurons to work. So I think it starts here. ATP binds to the myosin head, and what that does, this is really important, is it separates the myosin head from the actin filament. So the ATP just attaching to the myosin head does something like this. Here is the myosin head and the actin filament. When the ATP attaches, here's some ATP, this little eraser. Basically what happens is the myosin head just falls away from the actin. So this is still ATP, so no energy is being released yet. It's just basically the ATP comes in, myosin head's held on to the actin filament, doing its tug-of-war thing. And in order for the myosin head to leave the actin, the ATP has to come in order for it to get that same spot, right? Think about it that way. Then what happens is ATP, being an energy source, what you have to do is you have to break the phosphate bond. ADP is like a battery discharge. And you can see what happens here is ATP is for, I know those of you who have Bio 1 know all about this, but if you haven't had Bio 1 yet, ATP is basically a adenosine with 3 phosphates. There's like 3 little circles here indicating 3 phosphates. ADP is adenosine diphosphate. There's only 2 phosphates left, and a phosphate goes off. That phosphate breaking off basically is a transfer of energy. So what happens when ATP is converted to ADP is the myosin head cocks, right? So the ATP coming in allows myosin to leave the actin, and then ADP forming, the myosin head actually energizes or kind of like cocks. I kind of think of it like you know, those old Western guns, you know, like that have a hammer and you pull the hammer back, and, you know, it's like you're using energy to kind of pull the hammer back on a rifle or like a little 44 or Colt or something. But so in this stage here, ADP is formed, and the myosin head kind of energizes or cocks, so it's ready to go. Then what happens is some kind of a cue will happen here, and that cue will allow the myosin head to jump across and grab onto or bind to the actin filament. And so the cross bridging happens because myosin head's been energized and now can jump up and do that. Then what happens is the myosin head pulls it dissipates its energy by pulling on the actin filament, and the ADP is released here. But the thing here is the myosin head is chemically bound to the actin filament. It's stuck there. It had it grabbed onto it here like this, and grabbed on, pulled, but the myosin head is still stuck onto the actin until ATP comes in again and releases it, right? So that's why I say that I think that this is probably a better way to think about how this process starts is ATP attaches to the myosin head releases from the actin. ATP is converted to ADP, energizing the myosin head, like cocking it. The myosin head can jump across, grab onto the actin cross-bridging, and then it pulls on the actin filament, releasing ATP. And but the myosin head is stuck or bound or cross-bridged to the actin until another ATP comes in. Make sense? Cool. So on the exam, I'll probably have a little table that asks you to describe what happens in sequence. I'll provide you with the events, just as I described, and the chemicals like the ATP, ADP, cross-bridging, whatever. You'll have to kind of put those together in sequence, right? Okay, so there is an interesting thing that happens when many animals die. You've probably seen this, like especially now with the deer breeding season kind of getting them, wrapping up or whatever. They're kind of into their breeding time. You see a lot of deer hit on the road, like driving out Placer or out towards Chico now, and you see that someone has hit a deer with the car. The deer's on the side of the road. It's dead, right? Well, what happens when an animal dies, like a deer being hit by a car, is within about, depending on the temperature, within about 6 to 8 hours after death, sometimes it's more like 8 to 12 hours cold, they get stiff, right? Like they kind of turn like into a rigid thing, and this is called rigor mortis. So rigor mortis. Rigor mortis is a term that we use for this stiffening of the muscles after death, and it literally means stiff or tough death. So rigor means difficult or tough or stiff, and mortis means dead. So 6 to 8 hours, when it's warm, or 8 to 12 hours when it's cooler, most animals will go into rigor mortis. And what rigor mortis is, this is a really good test question, by the way, is when you die, you're no longer breathing, and your heart's no longer circulating blood or oxygen. So you no longer are delivering oxygen to your cells, right, when you die. But all of the glucose and sugars and stuff are still at your cells, ready to be digested. So your cells basically go anaerobic and stop producing ATP, right? When you stop producing ATP, you can't separate the myosin head from the actin. So what happens is your muscles basically all end up with their myosin head stuck to the actin, right? So this takes some time, because you have some residual oxygen in your cells, and then your muscle will kind of go anaerobic, building up lactic acid to make ATP. But eventually what happens is, especially your skeletal muscles, because of residual nerve action, kind of the death throes, what happens is the myosin heads will end up binding to the actin filaments in most of your large muscles, and they get stuck. And so rigor mortis is, here's the test question, rigor mortis is your muscles are all stuck in this stage because you no longer have ATP available to separate the myosin head in the end. And then rigor mortis continues for usually a couple days until enzyme and microbial decay. So basically, what do you call that? You end up being digested by your own enzymes, and you end up being consumed by bacteria that are eating your body. The deer, not you, right? But anyway, is microbial decay and basically enzyme decay ends up breaking down that binding. The myosin gets broken down. Or you've probably maybe used on meat, what's called meat tenderizer. You can, if you have a steak that's really tough, sometimes it's because it has rigor mortis still going on. You can use meat tenderizer, which is an enzyme oftentimes to break down the myosin. Or you can use vinegar. Like if you're cooking steak and it's really tough, you can break down muscle fiber using vinegar as well, or lemon juice. So anyway, the basic idea is rigor mortis is when the myosin head is stuck to the actin filament, and it's because ATP is no longer available because the animal stopped breathing. So oxygen is no longer available. And then rigor mortis persists for usually several days until decomposition breaks the muscle down. Got it? Forensic pathologists like the coroner, etc., will use rigor mortis as a way to estimate time of death. Like if they find a human body that is still pliable, like you can pick the arm up and it still kind of flops around, you know that person died within, depending on the temperature, half a day or so. But usually by 12 hours or so, people will begin to go into rigor mortis. And there's different levels, like early rigor mortis, full rigor mortis. And then they died like within a few days. And then if it's post rigor mortis, for one, the body's usually bloated and smells. But if the muscles are pliable because there's no longer any fibers available, you know it's post-rigor mortis. So they'll use rigor mortis as a window of death when they're finding people's bodies. So it's kind of interesting study there. Okay, one more thing. What we haven't talked about is what triggers muscle contraction. So far, this is all automatic. When the myosin head reaches over and grabs onto the actin filament, it just keeps doing this crawling thing. And what stops the process is the sarcomere shortens. So the sarcomere basically gets shorter. Well, the reason why muscles can only contract is the myosin heads are pulling on the actin, and these Z lines end up getting closer together. What stops it is when these actin filaments get very close and they can't shorten anymore. That's what limits muscle contraction. Usually about 20% length. So muscles can only contract at max 20% of their length. We'll talk about that more on Tuesday with skeletons. But if you have a muscle that's 10 centimeters long, like maybe your bicep muscle is about 10 centimeters long, your bicep muscle can only contract less than 2 centimeters. But because you have a leverage advantage, you can move your arm a lot more than that. But if you really feel your bicep muscle, it's not really shortening that much. It's just like maybe 2 centimeters, maybe an inch. But that inch contraction is moving your arm. But the limit to muscle contraction is when these actin filaments bump into each other, basically, the muscle can't shorten anymore. Or when the myosin runs into the end of the Z line. Okay? So what I haven't discussed yet, which is an add-on here, is what cues muscle contraction. And that is your nervous system, your motor neurons transmit, usually the motor neurotransmitter is acetylcholine. And we'll describe nerve cells and acetylcholine neurotransmitters next week with nervous system evolution. But your nerve cells will send a message across your neuromuscular junction, and it will stimulate the muscle cell to contract. Namely, what goes on here is calcium is a key ion to stimulate muscle contraction. So your motor neurons will be stimulated by a calcium transfer. The acetylcholine is transferred across the neuromuscular junction, but calcium is what really triggers the muscle to contract, stimulated by the neuron. Okay, so I'm going to add something on here, and that is that there is a triggering protein that is what stimulates muscle contraction. On the actin, there is a protein, and I'm gonna go ahead and combine two names together. If any of you have ever had physiology or anatomy, I'm kind of simplifying this greatly. But there is a protein that covers the actin when the muscle is not in contraction. And this protein is called tropomyosin. So tropomyosin is kind of like a sheath protein that covers the actin film. And what the tropomyosin does is it prevents the myosin from grabbing onto the actin. So think of this. This is like using my props here. This is like tropomyosin here, covering the actin. And what that does is it prevents the myosin from grabbing onto the actin because it covers the binding spots. What triggers the myosin to be able to grab onto the actin is when the tropomyosin falls away, the myosin can grab, right? So tropomyosin is the covering protein. I'm going to kind of combine this with another protein that is called troponin. And I'm not going to worry about you knowing the difference here, but troponin is basically the binding site for calcium. I'll just go ahead and use dots here. But, so I'll go ahead and say that this is troponin. Okay, so what happens, so what we'll say is, on the exam, I'll just say troponin and tropomyosin they're basically combined together. Troponin is a binding site on the tropomyosin. It's basically a binding protein that is attached to the tropomyosin, and the tropomyosin is covering the actin. So troponin and tropomyosin kind of work together. What happens is when calcium is released into the muscle cell, calcium attaches to troponin, and then that separates the tropomyosin from actin. So we can simplify this as one step and just say calcium triggers troponin tropomyosin to separate from the actin. And when the tropomyosin separates from the actin, the myosin head jumps across, grabs onto the actin and pulls it. Got it? So it's calcium that stimulates muscle contraction by releasing tropomyosin indirectly, right? Got it? Okay, so I'm not going to worry about you defining tropomyosin versus troponin. They're kind of related. And what it is calcium is our primary driver of this. Colors here. Let me use black here. Calcium binds into these troponin sites, and the tropomyosin can actually separate from the actin, and muscle contraction starts. Okay, so where does this happen? If I were to say where the calcium binds to troponin releasing tropomyosin, it happens right here. So if we're using the clock as 12 o'clock, 3 o'clock, 6 o'clock, 9 o'clock, we're looking basically at like 1 o'clock is where calcium is released. And 2 o'clock would be tropomyosin separates from the actin. And what that does is it allows the myosin head jump across. Okay? So on the exam, I'll probably have a sequence of events asking you to start at maybe any given point. But probably the logical starting point would be calcium is released, separating tropomyosin from actin. What happens next? Right? Boom. The myosin head jumps across, pulls on the actin, the myosin head stuck to the actin, and then ATP comes in, separates the myosin head. ATP is converted to ADP, energizing the myosin head. And the process continues as long as there's calcium present. And to stop muscle contraction, basically calcium has to be pulled out of the muscle cells. There you go. Sound good? Okay, cool. So let's go ahead and take another break. And a little, like a longish 10-minute break so I can get specimens out. Today's lab, pretty straightforward. It's just big specimens, so no microscope work really. But we're going to look at the oddballs and the kind of our goals today. I guess there is some microscope work. I'm sorry, I'm kidding. We do have a couple specimens to look at on microscope. So, but it's not too hard. It's only take about 4 specimens. So take a 10-minute break, and we'll get on with that. And what I want to get a picture of this.